Our next speaker is Julie Keister. Julie is a biological oceanographer with the University of Washington and Washington Sea Grant. She focuses on a variety of problems in biological oceanography related to phytoplankton and climate change, and her 5 minutes are dedicated to the effects of ocean acidification on the Puget Sound ecosystem.
Yes, so this is what I'll be talking about. So we all know a little bit about ocean acidification. It is caused by increased atmospheric carbon dioxide dissolving into the ocean and causing changes in the chemistry, which includes decrease in pH and a decreased availability of carbonate ions, which comes important in the physiology. An undergraduate made this slide for me, and I think it's awesome. So the Pacific Northwest is a really interesting place to study the effects of ocean acidification because we experience very extreme ranges of pH naturally, which are being exacerbated by anthropogenic increases. So for instance, in this transect, let me please get this, oh no, top one. That'll go away, right? This transect from the coast into Hood Canal, I'm just going to talk over it, showing depth of pH along that transect shows some very low pH values in the deep waters, and here's Hood Canal showing pH levels of 7.4 and below, so keep that level in mind.
Next slide.
Thank you. Yes. So what does that have to do with organisms? Well, we probably all know about the calcifying organisms. These organisms that build calcium carbonate shells are in real trouble when the pH is that low because of the decreased availability of carbonate that they build their shells with. So here is one of our tastiest oysters, whoopsie daisy, and the effects of pH on their growth showing at 7.4, which is the bottom waters of Puget Sound already showing a very diminished growth of these organisms. So the poster child of ocean acidification is probably the pteropod. It's a marine snail, and this sequence of images shows the quality of the shell from high pH to low pH. So these are some very obvious problems associated with ocean acidification, but there's also a lot of organisms that are not so obviously affected, but the pH changes in the water column affect the pH within organisms. So any changes in pH balance can affect a variety of physiology, such as respiration, development, behavior, etc. But these are not quite so obvious. It's not obvious how the effects will cascade through populations, for instance. So what are we doing in my lab? I'm looking at a couple of these species that are not necessarily so obviously affected by ocean acidification. These are two species of zooplankton, a krill and a copepod. So why are we studying these? Well, these are really important in food webs. They're major prey of a bunch of different fish, including ones that we love to eat, and therefore fisheries managers are very interested in. I'm going to show you a little bit of data on the krill that we are studying in collaboration with the Ocean Acidification Lab at NOAA, Northwest Fisheries Science Center. They have a very sophisticated ocean acidification facility where we can tightly control temperature and pH and therefore examine the effects of organisms using these variables. And I'm studying the early life histories of these organisms because that's the very quickly developing and growing, very sensitive stages of life. And so we're asking a couple of questions that I'll show here. For instance, of the eggs that are spawned, how many of them successfully hatch under low pH conditions? And of those that hatch, how many develop and grow naturally at high versus low pH? These are some of the pH conditions that we're testing. We're testing everything from why am I not getting this?
Don't click that.
I'm not clicking that. Everything from the surface ocean pH to the deep ocean pH. That's not my slide. And I'm going to keep talking because I'm going to have to dance soon if I don't. But there we go. There's some conditions. Blue is not so stressful. The oranges and reds are fairly stressful expected future scenarios. And here are some results. So on the left is a plot of hatching success across that range of pH. And what we found is that there is no statistical difference among hatching success across these ranges. So the good news for krill is that their eggs are going to keep hatching just as well as usual under a range of pH scenarios. However, that is not true of their development. You see that from pH of 8 down to 7.1, there's a large decrease in the development, and that is indexing the growth rate. And there seems to be some sort of break between 7.7 and 7.4, and I can talk to you guys about why this looks so weird at 7.5 if you're really interested later. But 7.4, to remind you again, is the current deep water pH of parts of Puget Sound, and 7.7 is sort of mid-water depths. So already there are conditions that might be detrimental to these populations. So what's that mean to marine ecosystems? We don't know yet. But thank you very much.